For 50 years, neuroscientists have used, well, one specific chemical to cut the power to the body's fight-or-flight nerves. I mean, they just inject it, the nerves die, and they record the results. But a newly published paper has just revealed that this trusted tool has been living a double life. It's basically been hiding a massive, invisible second identity right under our microscopes.
Yeah, it really forces us to ask a thrilling, but also, pretty terrifying question.
Yeah.
What if a fundamental piece of our scientific toolkit has been doing something entirely different than what we thought for half a century? How many assumptions would we have to just completely rewrite?
Right, exactly. So today we are unpacking a biological whodunit that attempts to answer exactly that for you. We are looking at a July 2026 paper published in the journal Neuron by you and their team. The title is dense, but it's really fascinating. It's called 6-hydroxydopamine promotes anti-tumor immunity through macrophage remodeling beyond sympathetic ablation.
Right, which is a mouthful. But our mission for this deep dive is to decode how a famous neurotoxin, which was brought into this experiment purely to act as, a set of biological wire cutters, accidentally turned out to be a secret weapon for the immune system against breast cancer.
It's wild. But we need to start the scene of the crime, right? The target of this neurotoxin is the sympathetic nervous system. We always hear the phrase fight or flight, but how does that actually connect to a tumor growing in breast tissue?
Well, it comes down to how tumors survive. You have to think of a tumor as essentially a rogue organ. To grow, it needs a blood supply, it needs nutrients, and a supportive environment. The sympathetic nervous system is the network that manages your body's stress response, flooding the system with neurotransmitters like noradrenaline.
Okay, so stress chemicals.
Exactly.
Yeah.
And there has been a long-standing hypothesis in oncology that chronic stress, and specifically these sympathetic nerves, might act like a supply line for cancer. The idea is that the nerves literally plug into the tumor microenvironment and feed it growth signals.
Ah, I see. So the logical scientific response is just cut the supply line, see if the tumor starves or stops growing.
Yeah, that was the foundational goal for the researchers here. They wanted to definitively sever these sympathetic nerves in mice with breast cancer to see if the tumors would shrink. And to ensure their results were really robust, they used 3 distinct methods to cut those nerves, testing them across 2 different breast cancer models, which are known as PIMT and 4T1.
So 2 different models, just to be sure it's not a fluke. Let's talk about how they actually cut these nerves, because the methods themselves highlight how clever biological research has to be. What was the first approach?
So method 1 was the industry standard. It's a chemical called 6-hydroxydopamine, or 6-OHDA. It's a neurotoxin. But the way it works is brilliant. Molecularly, it looks almost identical to dopamine and noradrenaline.
Oh, so it's a mimic.
Right. Sympathetic nerves use a specific cellular doorway, the norepinephrine transporter, to pull in normal neurotransmitters from their surroundings.
Yeah.
Because 6-OHDA is such a close mimic, the nerve mistakes it for food and actively pulls it inside.
Wow. It's literally a Trojan horse. The nerve willingly invites its own destruction.
Exactly. And once it's through the door, 6-OHDA triggers massive oxidative stress. It essentially rusts the cell from the inside out until the neuron dies. It's been the go-to chemical for sympathetic ablation since, the 1970s.
But a chemical is messy, right? I mean, it's circulating everywhere in the body. If I'm a scientist trying to prove to you that only the nerve is the issue, I'd want something far more precise to back up my chemical data.
Yeah, which brings us to the second method, genetic engineering. They used mice engineered so that their sympathetic neurons and absolutely no other cells in their bodies expressed a specific lethal vulnerability.
Wait, how does that work?
Well, in one model, called the DTR model, they programmed the nerves to create a receptor for diphtheria toxin. In another model, the DTA model, they programmed the nerves to actually manufacture the diphtheria toxin internally.
Let me make sure I'm visualizing this right. They went into the DNA and programmed the sympathetic nerves to essentially build a self-destruct button, leaving every other cell in the mouse completely untouched.
Yep. It's an incredibly elegant way to eliminate the nerves without flooding the system with a toxic chemical. And then, for absolute certainty, they used a third method, which was physical surgery. They went in with microscopic surgical tools and physically severed the nerve bundles, leading directly to the mammary glands.
Okay, so we have a chemical Trojan horse, a genetic self-destruct button, and literal microscopic scissors. 3 completely different ways to cut the power to the tumor. And here is where the paper introduces the massive plot twist.
Oh, it's a huge twist.
Right. The power goes out every single time. All 3 methods successfully destroyed the sympathetic nerves, but only the mice that got the chemical, the 6-OHDA, actually saw their tumor shrink.
Yeah, it's the defining anomaly of the study. The surgical and genetic methods were actually more thorough at wiping out the nerves than the chemical was.
Yeah.
The power was definitively cut. But the tumors in those mice just kept growing as if nothing happened.
Which implies the power outage isn't what's hurting the tumor at all. Like, if we have 3 ways to cut the power to a building and the power goes out every time, but the alarm only rings when we use one specific pair of wire cutters, then the power outage isn't causing the alarm. It's the wire cutters themselves doing something extra.
That's a perfect analogy.
Yeah.
The researchers recognized that immediately, and they proved it by using a special mutant mouse model called the SLC6A2 mutant. In these mutant mice, that cellular doorway we talked about earlier, the norepinephrine transporter, is physically broken.
So the 6-OHDA Trojan horse is completely locked out. It can't get inside the nerve to kill it.
Right. The sympathetic nerves in those mutant mice survived the 6-OHDA treatment perfectly fine. But when the researchers gave those mice the chemical, the breast cancer tumor still shrank. The anti-tumor effect had absolutely nothing to do with the missing nerves.
Okay, we are a few minutes in, so let's hit our first recap checkpoint. I want to make sure the foundation of this mystery is locked in for you listening. 1, the operating theory was that killing stressed nerves would stop cancer. 2, researchers destroyed the nerves 3 ways, but only the 6-OHDA chemical actually shrank the tumor. And 3, even when they used mutant mice where the chemical couldn't kill the nerves, it still shrank the tumor.
Exactly.
So this famous nerve-killing neurotoxin has a second, completely separate job. And I think this highlights something fascinating about the scientific process itself. You can get the expected result for completely the wrong reason, and the entire field might believe it for decades until someone spots the anomaly.
Yeah, anomalies are the breadcrumbs that lead to paradigm shifts. The challenge for the team now was figuring out what that second job actually was.
Right, so if it's not killing nerves, what is it doing? The most straightforward guess would be that it's just acting like chemotherapy. Is it just poisoning the cancer cells directly?
That was their first assumption. To test it, they needed to measure the life cycle of the tumor cells. If 6-OHDA acts like chemotherapy, you would expect to see a massive spike in dead cells, or a sharp drop in cells dividing and creating new ones. They ran specific chemical assays to measure both death rates and birth rates.
And what did they find?
Neither metric changed. The chemical wasn't killing the cancer cells directly, and it wasn't stopping them from dividing. They also checked the blood to see if it was altering systemic hormones like estrogen, which fuel breast cancer. Again, the levels were perfectly normal.
Wow, so it's not poisoning the tumor, it's not starving it of hormones, and it's not cutting the nerves. The list of suspects is getting very short.
Which means the cancer cells themselves are reacting to the chemical in a completely unexpected way. The researchers discovered that 6-OHDA triggers a very specific ancient alarm system inside the tumor cells known as the cGAS pathway.
Okay, cGAS, how does that work? What is it actually looking for?
Think of cGAS like a highly sensitive smoke detector, but instead of looking for smoke, it's looking for misplaced DNA. You have to look at this from an evolutionary perspective. For billions of years, the biggest threat to a single cell was a virus.
Right, because viruses inject their DNA.
Exactly. A virus works by injecting its own genetic code, its DNA, into the fluid of a host cell, the cytosol. So cells evolved cGAS as a chemical tripwire. In a healthy cell, all the DNA is locked away safely inside the nucleus. If the cGAS sensor bumps into free-floating DNA in the main fluid of the cell, it assumes the cell has been breached by a virus.
But wait, 6-OHDA isn't a virus?
No, it's a chemical stressor. When the tumor cell absorbs it, the 6-OHDA causes massive internal oxidative damage. It's like setting off a grenade inside a library. Books are going to fly everywhere. In the cell, pieces of its own DNA get blown out of the nucleus and end up floating in the fluid.
Oh, and the cGAS sensor bumps into them.
Right, bumps into its own DNA, thinks the cell is under attack, and pulls the fire alarm.
Okay, so when a building's fire alarm goes off, doors lock, sprinklers turn on, what does the cancer cell do when its cGAS alarm trips?
It synthesizes and releases a massive distress signal, a protein called interferon-beta, or IFN-β.
Interferon-beta. I've heard of interferons in the context of getting sick, like fighting off a flu.
Yeah, they are incredibly potent immune activators. They literally, interfere with viral replication, hence the name, but they also act as a glaring distress flare. Tumors normally survive by cloaking themselves. They create a dark, immunosuppressive microenvironment, so the body's white blood cells just float right past them, completely unaware of the danger.
Okay, so if I want to visualize this, it's like the 6-OHDA is a pepper that makes the cancer cells sneeze out this IFN-β flare, lighting up that dark environment.
That's a great image.
Yeah.
And that flare initiates a radical transformation in the immune system. This is where we see the domino effect begin. Floating in your bloodstream right now are white blood cells called monocytes, specifically ones with a marker called I6C2. They are essentially raw, unspecialized recruits on patrol.
Right, just looking for trouble.
Exactly. When they detect that interferon-beta signal, they swarm into the tumor tissue.
And the paper talks about them remodeling into, ISG-positive macrophages. What does that actually mean? How does a cell remodel itself?
So when those monocytes bathe in the interferon flare, the chemical signal actually penetrates down to their DNA. It turns on specific genes, interferon-stimulated genes, which is what ISG stands for.
Got it.
It fundamentally rewrites the cell's identity and function. A standard macrophage is basically a cellular garbage collector. The name literally means big ear. It wanders around eating dead tissue and cleaning up debris. But this new remodeled macrophage, the ISG-positive one, becomes a highly specialized, heavily armed commander.
So what makes it a commander? What new tools does it get from this genetic rewrite?
Well, it starts expressing high levels of specific markers like CD64 and SCAL1. But the most crucial new tool is a surface protein called MHC-Tachnite. If the macrophage is a commander, MHC2 is how it holds up a mugshot of the enemy.
A mugshot.
Yeah, it scavenges pieces of the dead cancer cells, presents them on its surface using this MHC-TO protein, and shows the rest of the immune system exactly what the target looks like.
Wait, so it's holding up a mugshot, but the tumor is huge and the body is vast. It needs to actually summon the specific assassins to come look at the mugshot. How does it physically call them in?
It creates a chemokine gradient. Chemokines are chemical attractants. These remodeled commanders start pumping them out into the surrounding tissue. As these molecules diffuse outward, they create a chemical trail. The closer you get to the macrophage, the stronger the chemical scent becomes.
And who is following that scent? Who are the assassins?
The Th1 cells, type 1T helper cells. These are the specialized heavy hitters of the adaptive immune system. You can identify them by a specific transcription factor called TBET.
Okay, so the Th1 cells show up.
Right. They follow the chemical trail, read the mugshot presented by the macrophage, and then unleash their own weapon, which is interferon gamma.
Another interferon.
Yes, but this one acts as a direct execution order. Interferon gamma directly halts tumor growth, and crucially, it feeds back into the macrophages, making them even more aggressive. It creates a completely self-sustaining, lethal loop of anti-tumor immunity. The macrophage called the Th1 cell. The Th1 cell attacks the tumor and empowers the macrophage, which calls more Th1 cells.
That is an incredible biological machine. But tumors are notoriously stubborn, right? They spend all their energy actively suppressing the immune system. Why would the tumor just ignore this whole process? Like, how do the researchers know this specific chain reaction from the macrophage to the Th1 cell is actually the thing shrinking the tumor and not just some background noise?
To prove it, they had to break the chain on purpose. They tested the structural integrity of this pathway using a drug called BLZ-945.
BLZ-945. How does that drug work?
So macrophages are powerful, but they require a constant chemical survival signal to stay alive in a tissue. They rely on a receptor called CSF1R. It's essentially a dead man's switch. If that receptor isn't constantly stimulated, the macrophage undergoes programmed cell death. BLZ-945 blocks that receptor. So if you give a mouse this drug, you effectively wipe out the entire macrophage population in the tumor.
You eliminate the commanders.
Exactly. They triggered the 6-OHDA flare, but simultaneously wiped out the macrophages. The result was stark. The Th1 assassin cells never showed up, and the 6-OHDA completely lost its ability to fight the cancer. Without the commanders to lay down that chemical trail and hold up the mugshot, the whole attack fell apart.
Wow. And they even tested the other end of the chain too, didn't they?
They did. They used genetically modified mice that entirely lacked CD4 T cells, which is the specific family that Th1 cells belong to. And again, without the Th1 cells, the 6-OHDA was useless. The tumor just grew. It proves with absolute certainty that this drug is working through this highly specific pathway, the ISG-positive macrophage recruiting the Th1 cell.
Okay, this is the perfect moment for our second summary checkpoint. Let's make sure the dominoes of this mechanism are crystal clear. Domino 1, the 6-OHDA chemical, causes massive internal damage to the cancer cell, tripping the cGAS smoke detector, which forces the tumor to fire an interferon-beta flare.
Correct.
Domino 2, that flare, attracts circulating monocytes with those IL-6C2 markers and remodels them into heavy-duty macrophage commanders with the ISG-positive genes. Domino 3, these commanders hold up an MHC2I mugshot of the cancer and lay down a chemical trail to recruit Th1 cells. And domino for the Th1 cells attack the tumor with interferon-gamma and empower the macrophages, creating a lethal loop.
That's a perfect summary.
And if you break any link in that chain, remove the commanders with BLZ-945, or remove the assassins by knocking out CD4 cells, the whole thing just stops.
It really is a brilliantly complex mechanism, and the experiments prove it beautifully. But there's a massive caveat.
Yeah, in biology, there is always a caveat.
We mentioned earlier that 6-OHDA worked in the PIMT and 4T1 breast cancer models. But when the researchers tested a third model, called E0771, the immune cascade bounced right off. It did absolutely nothing.
Wait, why would it fail? If this macrophage Th1 axis is so potent, why did it hit a brick wall in that specific tumor?
Well, it comes down to understanding the baseline environment of the tumor itself. Tumors are chaotic. Some grow so rapidly that they outstrip their own blood supply. The centers of the tumors become hypoxic, literally suffocating from a lack of oxygen.
Okay, so they're choking.
Right. And this constant suffocating environment causes massive internal damage to the cells, which means DNA is constantly leaking, which means the cGAS sensor is constantly tripping.
Oh, so the E0771 tumor is existing in a permanent state of panic.
Exactly. The E0771 tumor cells are intrinsically stressed. Because they are constantly tripping their own alarms, they're already producing a massively high baseline level of interferon-beta on their own just by existing.
Ah, I see.
So when the researchers introduced 6-OHDA to those specific cells, it couldn't push the alarm any louder. The local immune system was already saturated. It was completely maxed out on that specific pathway.
That makes perfect sense. It's like if the fire alarm is already blaring at maximum volume, lighting a match in the corner isn't going to change the response. You just can't wait up a biological pathway that is already wide awake.
That's it exactly. This tells us that 6-OHDA's hidden immune trick has a ceiling. It only works on tumors that have a low baseline of interferon. It's a powerful tool, but it is highly context dependent.
Okay, so pulling all of this together for you listening, we've gone from nerve-cutting chemicals to smoke detectors to immune commanders. Why does this molecular deep dive matter to someone listening right now?
Well, it matters because it forces a massive reevaluation of medical history. For half a century, the global scientific community has used 6-OHDA under the strict assumption that it only did one thing: kill sympathetic nerves. Think about the thousands of papers written on stress, Parkinson's disease, nervous system development, and cancer that relied on that fundamental assumption.
It's staggering to think about, like, how many researchers over the last 50 years saw a tumor shrink or a disease change and wrote a paper attributing it entirely to missing nerves when in reality they had accidentally triggered this massive hidden immune response.
Exactly. We have to look at decades of past data through an entirely new lens. But looking forward, it also gives us a brilliant blueprint. I mean, 6-OHDA is a neurotoxin. We can't just give it to human cancer patients. It would destroy their nervous system.
Yeah, right. That would be bad.
Very bad. But now that we know the exact mechanism tripping cGAS to remodel macrophages, we can hunt for safe, non-toxic drugs that push this exact same domino. If we find a drug that triggers this self-sustaining immune loop without the neurotoxicity, it could completely revolutionize immunotherapy for solid tumors.
We started today looking at what seemed like a straightforward tool, a chemical wire cutter for the nervous system. By pulling at a single loose thread, an anomaly where the tumor shrank, even when the nerves survived, you and his team uncovered a hidden trapdoor in our biology, a potent, self-sustaining immune feedback loop hiding right beneath the surface.
Yeah, it is a masterclass in the scientific method. It shows why we must never take our tools for granted and why we have to let the data lead, even when it completely contradicts 50 years of textbook assumptions.
I want to leave you with a thought to mull over today. If a famous, highly studied chemical used confidently by scientists since the 1970s has been secretly activating the immune system this entire time without anyone noticing what other established facts in science, or even trusty tools in your own daily life and profession, are hiding a massive, invisible second function, are we always measuring what we think we're measuring?
That's a really profound question to take with you.
Thank you for taking this deep dive into the microscopic world with us. We'll catch you next time.